Hi, my dear students, and welcome to Physics Wala, an incredible platform. And I am your zoology teacher, and my name is Diksha Sharma. So, I'll be teaching you zoology if you enter to this batch, that is Yakeen English channel. Most of you must have seen me somewhere, either in the Umeed or in the different batches, or in fact in the PW English channel. So, let's get started with a very small topic, and that is nerve impulse. How the nerve impulse is, generated, how it is conducting, and then how it transmits. So let's get started with this topic. So let's get started with the topic that is nerve impulse. So you all must have heard of nerve impulse, right? Or impulse. It's not that pulse that you detect it here through the wrist, and neither this is the pulse toffee that you eat. Yeah, there is a candy named pulse also. So nerve impulse, what is it? We say that neurons are excitable cells. What are excitable cells? That can generate the electric current, or they can also detect the electric current and they can respond to that, right? So nerve impulse is an electrobiochemical phenomenon. It's just like an electricity or an electric current that is flowing across the neurons membrane, right? So what is a nerve impulse? We say that our neurons are excitable cells. So it's basically the electrical signal. It's what? It's the electrical signal that transmits or flows or travels through a nerve membrane. Through a nerve membrane. So this is what excitable means, that any cell is excitable if it can detect or it can generate this electric current, right? So we say that if this is your neuronal cell, if I say it is a cell membrane, it's a neuronal cell membrane. It's a neuronal cell membrane. We say that a current is flowing through this, neuronal cell membrane. Then why or how does this electric current moves, and what is that phenomenon that leads to the formation of this current? So there is one thing that are responsible for generation of this current is a stimulus. So whenever a stimulus arises, the nerve cells gets excited. They started producing electric current. So now, yes, okay, fine. The stimulus arise and they got excited. Now, what is the thing that leads to the change in charge? So that are the ions. So this cell membrane leads the flow of ions. So for the flow of ions, we need a passage because the cell membrane is made up of lipid. If I'm saying only if the ions are moving across the membrane, the charge developed, and that leads to the formation of electrical signal. So if I say the membrane is lipid and lipids are insulator, they will not allow the formation of this charge. Then how will the charge will be formed? So for that, we need we have certain ion channels. We have certain ion channels. For example, this is sodium ion channel. So when this open, that leads to the movement of sodium ions, right? So what are these? These are ion channels. So ion channels are nothing but these are just proteins. These are the doors made up of protein across the cell membrane, and that will allow the movement of the ions, leading to the change in the charge and hence the voltage. And then what we say, not in fact the voltage, you always detect the electric current in millivolts, just like that. So that will lead to the formation of a kind of an electrical signal. Now these ion channels, they are of different types. These ion channels, they are of different types. First of all, what are these? These are made up of protein, and they allow the passage of ions. Allow the passage of ions. Fine. So now what happened here, guys? Ion channels, they are of different kinds depending upon what type of ions they are transporting. For example, this one is only helping in the transport of sodium ions. So this is a sodium ion channel, right? So there are different kinds of channels. Some channels are leaky channels. They are open all the time. right? They randomly, they can open, they can close, just like this one. These are normal ion channels, like sodium ion channel. So sodium ion channel will only be leading to the transport of sodium ions, and potassium ion channel will only lead to the the transport of the potassium ions. Then we have certain channels which can only open and close according to the stimulus, right? If that thing will come, only then they will open. For example, if I say there are ligand gated ion channels. So ligand gated ion channels need to have certain chemicals that will open them. For example, if I say this is the sodium ion channel, but this is not the leaky one, this is not a normal sodium ion channel, this is the one which is a ligand gated. For example, if to this ion ACH, what is ACH? A neurotransmitter. If to this channel the ACH binds, only then this will open and lead to the transport of sodium ions. So they will open and close according to the chemical that binds. According to the chemical that binds. Right? And then we have the voltage gated ion channels. Voltage gated. Now, what are these? If ever there is a change in the potential difference that is same you study in the physics, right? So if there is any change in the potential difference across the cell membrane of neuron, only then these ion channels will open. So they open and close with respect to WRT. Diksha ma'am always write WRT, that is with respect to, right? With respect to the change in potential difference. Potential difference. Fine. So like we have sodium ion voltage gated channel. So as you can see, what is that common thing? The common thing is everyone is transporting sodium. There can be sodium ion channels in the leaky, there can be sodium ion channels in the ligand, there can be sodium ion channels in the voltage. The only difference is what is the kind of stimulus that open and close it. For example, for opening this kind of a channel, you need to have a ligand, a chemical. To open this kind of a channel, you need change in the potential difference. To open this kind, they open and close randomly. You do not need a lot of stimulus for that, right? And sometimes, yes, stimulus is needed, but normally they are always in the open state. Fine. All right. So that's about the ion channels that leads to the transport of ions and which lead to change in the potential difference, change in the charge, and hence that will lead to the production of electric current. Now this is about the theory. Now how actually the generation of impulse takes place. So for that you need to understand one very important topic, that is, okay, RMP. What is RMP? Resting membrane potential. What's the full form of RMP? Guys, resting membrane potential. Resting membrane potential. Now what is this? So whenever our neuron is resting, no stimulus have been arised, and it is not generating any impulse. At that time, the situation in which the neuron is sitting silently doing nothing is known as resting membrane potential. And you must have thought, ma'am, but that's a potential. Very true. The resting membrane potential is the potential of your nerve membrane when it is at resting state. Again, I'm telling you, RMP's resting membrane potential, it's the potential difference across the cell membrane. What cell? Neuron. When it is doing nothing, when it is just resting, right? So what is resting membrane potential? It is the potential across the neuronal membrane when it is at rest. When it is not doing anything, it is completely at rest, right? So let's see what is that. For example, again, this is your neuronal cell membrane. And if I say this is the outside and this is the inside. So we are not drawing the entire cell. I have just drawn one axonal membrane or membrane cell membrane of axon, right? So we say that outside always, that means outside the cell, always the concentration of sodium ions is higher, and inside always potassium ions are higher, right? So we always say that whenever we are talking about a cell, outside the concentration of sodium ions, that in your body fluids and in the interstitial fluid. What is an interstitial fluid? The fluid present in between the tissues, right? And the potassium ions, they are higher inside the cell. Okay. Now, what happened during resting state? Our entire membrane is more negatively inside and positively outside. We say that during resting state, our cell membrane have a certain charge difference. Inside is more negative and outside is more positive. As a result, across the membrane, if you will see, the difference in the voltage, or we call it as difference, or you call it as potential difference, then inside it is more negative. So you call it as, it is how much negative? Inside is minus 70 millivolt. Inside is minus 70 millivolt. That's the potential difference across the cell membrane. So we say inside it is more negative, outside it is more positive. Now, what are the factors that lead to this minus 70 millivolt situation? Why it is always minus 70 millivolt? Or why we say that sodium concentration is higher outside and potassium is higher inside? Why so? Why so this happen? There are three reasons. I say sodium is always higher outside, potassium is always higher inside, and the potential difference across the cell membrane is minus 70 millivolt, right? Why is it happening? Why is it happening? There are three reasons. How many reasons, guys? Three reasons. Let's check them out. First reason is differential permeability. Differential permeability of ions. What does it mean? Differential permeability means there is a difference in the permeability. Permeability means anything that is allowed to move or not. For example, if I say sodium ion can run faster, potassium ion runs slower across the membrane, then we will say they have different types of permeability. Permeability, that means allowance to move. Fine. So why does this happen? For example, if I say this is a cell membrane, it has more number of potassium ion channels than sodium ion channel. If I say there are more number of potassium ion channels than sodium ion channels, then what will happen? Just think, have some brainstorming. Just use it, otherwise it will go bad, So if I say there are more potassium ion channels than sodium ion channels, then what will happen? And we always say sodium is higher outside and potassium is higher inside. And you have also studied somewhere in the botany that things always move from high partial pressure to low partial pressure, or we say in case of osmosis, it always moves from high concentration to low concentration. In zoology, you always study high partial pressure to low partial pressure, and in botany, according to osmosis, the things always move from high concentration to low concentration. So how will these things move? If I say this is one sodium ion channel, only one sodium ion will enter, right? Whereas more potassium ions will move outside. So as you can see here, two potassium ions have move outside. So how many positive charge the outer part of a membrane have? 2. Now only one sodium ion have entered inside. Now how much charge the inside membrane have? 1. So if we will create a difference, the inside one is at loss or at negative side of 1 as comparison to outside. So we say that outside is positive, inside is negative. Why? Because there is more efflux. More efflux. Efflux means moving outside. Efflux means moving outside. More efflux of potassium ions than influx of sodium ions. As a result, the outside is more positive and inside is more negative. This is the reason why outside is positive and inside is more negative. This is the first reason. Okay, now let's look about the second reason. The second reason is sodium potassium ATPase pump. Sodium potassium ATPase pump. What are pumps? These are not the hand pumps that will pump water from the groundwater and give it to you. No, 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 no. It's not like that. Pumps are what? For example, if this is the membrane, in the membrane, certain proteins are present. What are present? Certain proteins are present. These proteins, they act as ATPase. How do they act as? ATPase. And they have seats. You heard it right? Seat. Seat to sit on. And these seats are not for you to sit on. These seats are for the ions to sit on. Now, what it will do? It will pump three sodium ions outside and two potassium ions inside by using one ATP molecule. You know, this ATP with the help of this enzyme will be converted into ADP and IP. And you all know ATP is the energy currency. When IP is lost, it will give energy and it will lead to the functioning of the pump. And what will pump do? Pump will, for example, if this is a pump, okay, this is the inside, this is the outside. This is the inside, or you just take it this manner. This is outside, this is inside. On this side, which is facing towards me, the three sodium ions will sit here and two potassium ions will sit here. Now ATP will come the energy and this pump will move like this. As a result, the three sodium ions will be pumped outside and two potassium ions will be taken inside. So there is a benefit again. Three sodium ions are moving outside. 1, 2, 3. Only two potassium ions have been put inside. So again, the inside is more negative, outside is more positive. So this is outside and this is inside. Fine. Another thing, you also have a benefit of something. We say that the concentration of sodium ion is always higher outside. So these pumps will make sure the sodium ions should be higher outside and potassium ions should be higher inside, right? Now the third reason, let me just use another page for that. Okay, what's the third reasons? The anions. What are anions? Anions are negatively charged. They are negatively charged and they are larger in size. They are larger in size than the cations like sodium ion and potassium ion. Now what happened? Imagine this is the neuron. Okay, and this is the membrane of neuron. And we say that in the membrane outside there is always positive charge and inside there is negative charge. So one such reason is differential permeability, second the pump, and third anions. Anions do not have the channels to move, right? Anions are trapped inside your cell. Anions are trapped inside your cells and they do not have any channels to move outside. So what happens whenever there is accumulation of positive charge, whenever there is accumulation of positive charge to the outside, they will get attracted and they will accumulate towards the membrane like that. Anions are always trapped for just like a magnet. Like for example, if you're sitting in a room and I give the magnetic charge to the entire walls and you put some iron thing inside, or if I say I give charge I make your walls like a magnet and I put a one iron thing inside the room, then when that magnet or when the walls are converted into magnet, that iron thing will get attracted toward the wall. Just like that. So there also, when the positive charge accumulate outside, the anions, they will move towards the membrane. As a result, there is a creation of the charge difference. The charge difference has been created along the membrane and hence inside is more negative and outside is positive, right? So we say that the anions, we say that these anions are negatively charged. They do not, let me just write it with another color, they do not have any anion channels for their movement. So they get attracted towards positively charged anions present outside the membrane, creating a charge difference. Creating a charge difference across a membrane, across the membrane. So this is how there is a negative charge towards inside and positive outside. Let's see what NCERT has to say. Okay. So neurons are excitable cells because, okay, that might not be clear to you. Let's increase the font. Okay, that's my way of teaching, guys. I always use NCERT clips so that you will not face difficulty later on, because you know there are certain sentences in the NCERT that are way difficult for you guys. So that's my habit to give the NCERT. Okay, so those who are there for the first time, who have not seen any of my lectures before, they must be knowing that. But those who are new here, you're a new student, and so that's the way how your ma'am teaches, right? Okay. So do you know why the membrane of a neuron is polarized? So as I said, there is a charge difference. You can also call it as polarized. Different types of ion channels are present on the neuronal membrane. As I've told you, there are ion channels present. These ion channels are selectively permeable to different ions. So selectively permeability means differential permeability means different types of permeability. So because they're selecting things, so you can also call it as selective permeability. Okay. When a neuron is not conducting any impulse, that is resting, the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. This is what we have done. So whenever it is at resting membrane, potential is there is differential permeability, there are more number of potassium ions, that means there is more efflux of potassium ion, and this is what you call it as permeability also. Then similarly, the membrane is impermeable to negatively charged proteins present in the exoplasm. Consequently, the exoplasm inside the axon contain high concentration of potassium ion and negatively charged proteins and low concentration of the sodium ion. So we say that here that the anions, what are these anions? They are basically present in the form of proteins. So anions, what anions can be there in the cytoplasm? They all are present in the form of what? Proteins. These are basically what? These are basically all proteins, right? Okay, so as a result, they are always present towards inside, and they in the next line they have said that inside the potassium ions are higher and outside sodium ions are higher. All right. In contrast, the fluid outside the axon contain a low concentration of potassium ion, a higher concentration of sodium ion, and thus form a concentration gradient. So there is a gradient form. That means we always say sodium is higher outside, potassium higher inside. These ionic gradient across the resting membrane are maintained by the active transport of ions by the sodium-potassium pump, which transport 3 sodium outward and 2 potassium inside the cell. So here we have discussed, there I have told you also that whenever this pump works, it solves two purposes. One, it will definitely create negative charge inside. Second, it will make sure that sodium ion should be higher outside and potassium ion should be higher inside, right? Okay, next. All right, while its inner surface becomes negatively charged, therefore is polarized. The electrical potential difference across the resting membrane is called the resting potential, right? So this is all about why you have minus 70 millivolt, a polarized state, or you can say during the resting membrane, when the membrane of your neuronal cell membrane is completely in a rest form. And as I've told you, we are always talking about this membrane is a membrane of the neuron. And what part of a neuron? Axon, right? So it's basically what? Axon. So this cell membrane is always of neuron. You must write it. Why? Because after some time you'll get confused. Now you are hearing everything and you must be thinking, yes ma'am, yes ma'am, okay, okay, done, done, done. But always, always make good notes, guys. Okay. So here the cell membrane, and this cell membrane is of a neuron and basically of axon. So you also call it as exoplasm. What you call it as? Exoplasm, or sorry, you call it as exolemma. Exolemma, just cut the N here. Exolemma. Exolemma is a cell membrane, whereas exoplasm is a cytoplasm. Whereas exoplasm is a cytoplasm. All right. So that's all about why there is a resting membrane potential. Now let's move further and talk about how the nerve impulse is generated. Let's see. Now, guys, for example, this is a membrane and we say positive outside, negative inside, and this is RMP, and at RMP we have minus 70 millivolt. Okay, and we say this is outside, this is inside. Outside always the sodium concentration is higher, and inside always potassium concentration is higher. This is what we do. Okay, now moving further. So what we say here is that how does this electrical impulse is generated? First of all, always there is a stimulus that arises. Always, how does the neurons respond? What do you think? How does the neurons respond? For example, right now, if someone touches you, you're sitting in a table and someone touches you and asks you to talk to them, they will touch you. Okay, just like any of your friend is sitting behind you. So how that friend will call you? The friend will touch you, like they touches you here. Okay, and now suddenly you will move and say, what's the problem? Right? So just like that, that touching was a stimulus. So stimulus is something that is change in the environment, right? Stimulus is something that is change in the environment. Okay. So whenever the stimulus arises, there is a change in environment that leads to opening of sodium ion channels. Which ion channels open here, guys? Sodium ion channels. Now, if the sodium ion channels open and concentration of sodium is higher outside, sodium ions will enter inside. So the stimulus causes opening of sodium ion channels. Okay, so when the sodium ion channels open, the sodium ions are moving inside. One confusion that you always have, ma'am, but the positive charge is moving, then there should be, negative charge. No, the ions are moving, the electrons are not moving. Okay, so when the sodium ions move inside, the inside of a membrane will start it to become positively charged. As a result, from minus 55, it will first move to minus 55 millivolt. And this is a kind of threshold stimulus. What is a threshold stimulus? That this is a minimum stimulus that you need. This is a minimum stimulus that you need to reverse the polarity. This is a minimum stimulus that you need to reverse the polarity. For example, the sodium ions are entering. Now, as a result, one situation will come where the polarity got reversed. So once it has entered the minus 55 state, it will definitely go to this stage, which is of plus 30 millivolt. So that's why you call it as threshold stimulus. For example, if your membrane comes to around minus 40 millivolt, okay, it comes to around minus 40 millivolt and it stops there. So that means from there it will not go to plus 30 millivolt. Okay, why? Because that's not the threshold stimulus. We need minimum minus 55 millivolt to convert it into plus 30 millivolt, which is the entire opposite to it. That means we are reversing the polarity, and this stage from RMP to plus 30 millivolt, this one where polarity has been reversed, you call it as depolarization. Depolarization. And here you say the action has been done, so that is known as action potential. What do you call it as? Action potential. So now plus 30 millivolt have been, right, the membrane has become excited. This is why you call it as action potential. The action has been done, and what is that action? That means your membrane has got excited. Okay, now what will happen next? Now this will lead to the opening of potassium ion channels. And potassiums are higher inside, so they will move outside. So now there is opening of Due to plus 30 millivolt, there is opening of potassium ion channels. Now, once the potassium ion channels open, again the reversibility will occur. Inside will become negative, outside will become positive, and you call it as repolarization. You call it as repolarization, and here the potential difference is minus 70 millivolt. Now, you must be wondering, ma'am, this is also minus 70. Bam, this is also minus 70. Then why do we call it as our resting membrane potential? Why are we calling it as repolarization? We can also call it as from RMP it go to depolarization, then come back to RMP. Why this is not called as RMP? It is known as repolarization. The reason is here the ionic concentration gradient has also been disturbed. Here the ionic concentration gradient has also been disturbed. During repolarization, sodium ions are higher inside and potassium ions are higher outside. Yes, yes. Why? Because potassium have gone outside and sodium during depolarization they have entered inside. Now how will we correct it? Who is the one that can correct this? Who is the one that can throw 3 sodium ions outside and take up the 2 positive ions inside? That is your pump. That is your pump. So this pump will use one ATP, throw 3 sodium ions outside, put 2 potassium ions inside and get it back to minus 70 millivolt, right? And that will be your RMP. RMP, that is minus 70 millivolt. Okay, and this is how we get back here. Sometimes from minus 70, your this charge can also go to more negative, that is around minus 90 millivolt. Sometime if potassium ions have been opened for longer duration, they forget we have to move back. So potassium ion channels, they forget that we have to move back. So they're like open for long time interval. So that time, this stage is known as hyperpolarization. That stage is known as hyperpolarization. Even if it has entered into the hyperpolarization, it can get back to resting membrane potential by the similar manner. That means the use of ATPase, sodium potassium ATPase pump. So this is how the excitability is generated. When the stimulus arises, sodium ion channels open, that will reverse the polarity and you say this is now excitable. How it will move back? It will then lead to the opening of potassium ion channels, which will lead to repolarization, and then the pump will work and that will go to resting membrane potential, and this is how it go back to rest, right? So this is the position when there is a stimulus that arises on only this segment of neuron. For example, your friend touched you only here. So that means the stimulus was only here. Then how did your entire body with the uses of your skeletal muscles move like this? How does the impulse reach to your brain and then from brain to your muscles, and how did muscles, have taken up this impulse? Now that you call it as, my dear students, you call it as conduction of nerve impulse. What do you call it as? Conduction. See, one is generation of nerve impulse. That how with the help of stimulus at that one particular position, the resting membrane potential have been reversed and have caused depolarization. Now how in that segment where the depolarization has taken place by stimulus will move to the entire body. Now that is conduction. Okay, or we can say how, for example, if I say this is one neuron, okay, this is neuron. Imagine the stimulus have arrived here, then how does it will move towards the entire neuron? That is your conduction. This is your conduction. Right? Now, this neuron will end at these nopes. Okay. Now, how it will move from one neuron to another, and this you call it as transmission. Transmission. Okay. So, where stimulus arises, here you call it as generation of nerve impulse. After generation, how that depolarization wave moves towards the exolemma of the entire neuronal membrane, that is conduction, and how from one neuron it goes to another, that is transmission. So now we are going to talk about the conduction. Now for example, if the stimulus arises here, at this point, the A segment, we have divided the entire axonal membrane into 3 segments, A, B and C. Now if the stimulus arises here, this will lead to the second step. This will lead to the second step. In the first step, the stimulus arises. In the second step, what happened? The A segment, as you can see, it has undergone from minus 70 millivolt, it has entered to plus 30 millivolt, that is depolarization. So we say after stimulus, the A segment will undergo depolarization, right? Now, what will happen next? Now, what will happen here? There is a charge difference. As you can see, this is negative, positive, positive, negative. This charge difference will create a local circuit. And this local circuit will act as a stimulus for B segment. Right? Because for depolarization, definitely you need the stimulus. The first one was a different stimulus, like your friend touched you. How the second segment will take up the stimulus? It will generate its own stimulus. Now for that, what's the stimulus? The charge difference, the circuit that has been formed here. As a result, the B, okay, as a result, the B will now undergo depolarization. Okay, and this one in meantime is undergoing repolarization. Now again you can see there is a charge difference, right? And now that will act as a stimulus for the C segment, and now C will undergo depolarization. And this is how the nerve impulse will get conducted from one site of stimulus to the entire exolemma, right? Okay, now conduction is of two types. One is continuous, another is saltatory. Continuous is the one that we have seen right now. It is just moving continuously because there are no barriers. Right? There are no barriers, or there are not interruptions. There are not any interruptions. That's why it is moving on continuously in a smooth manner. Whereas in the saltatory, for example, if you are moving, okay, and there are certain big rocks that are present, or you can say the rocks of this 1 feet tall, they are present on some places. Then what will you do? You will step up, jump, and then move further, like that. Okay, so if you have no rocks in your path, then you can easily move like that continuously, just like that. Okay, so this is what happened in the continuous conduction. In continuous conduction, they are usually occurred in the unmyelinated nerve fibers. In unmyelinated nerve fibers, there is no myelin sheath. For example, this is how the nerve fiber is. There is no myelin sheath. There is no myelin sheath. So the impulse will move in the manner that we have just done right now in the ABCD segment form. Fine. Whereas in the saltatory, what happened? There is a nerve membrane, and there is the myelin sheath in places. So, because there is myelin sheath, it acts as an insulator. It will not allow the current to pass through. So, the current will move by jumping like this. That's why its name is saltatory conduction. Saltation means jumping, right? So, here nerve impulse jumps. So, what do you think? Which one is faster? This one is faster. Why? Because it is jumping, and it will also use less energy. Why? Because only few segments have been depolarized. So you don't need to use a lot of pumps. More pumps you use, more there will be ATP used, because pump always use ATP. For example, if I say there 1 ATP will be used, for this also 1 ATP will be used, for this segment 1 ATP will be used, because every segment is getting repolarized. For reaching RMP, they will be using pump. Fine. But here if on the B segment we have myelin sheath, then now how many ATPs will be used? 1 and 2. So less ATP will be used in saltatory conduction. Less ATP will be used in saltatory conduction. Okay, so this is how it can be conducting throughout the exolemma. Moving further, how does it is transmitted from one neuron to another? First of all, for transmission, we have synapses. What do we have, guys? Synapses. As we have already done here, synapses are of two types. One is a chemical synapse and another is the electrical synapse. Now, what's the difference? All right. So this is a very important topic, because from here the questions have been asked in the previous years. So you don't skip this one and pay attention here. Okay. So what happened, guys? For example, you have this one neuron. This is another neuron. And you need to send impulse from this neuron to this one. And this junction between the two is synapse. The junction between the two is synapse. Okay, now this neuron, which is present before the formation of synapse, is presynaptic neuron. This is presynaptic neuron. And this one is postsynaptic neuron. This is postsynaptic neuron. Similarly, this membrane will be known as presynaptic membrane. And this will be known as postsynaptic membrane. Okay. Now, the first neuron have nerve impulse that rises to this position in the form of the waves of depolarization and repolarization. Imagine here the nerve impulse have been entered. All right, here nerve impulse have been entered and we need to excite this one or inhibit this one, or we can say we have to send the information from this neuron to this one, from presynaptic to postsynaptic neuron. Now what will happen? So as you know, guys, here some small vesicles are present known as synaptic vesicles. So when the nerve impulse arises here, there is opening of calcium ion channels. There is opening of calcium ion channels in the presynaptic membrane. So in the presynaptic membrane, due to entry of the nerve impulse, there is opening of calcium ion channels and calcium ions will enter inside. And this will lead to fusion of the synaptic vesicle with the membrane, and this will release the neurotransmitter. What happened? When the nerve impulse arises, the calcium ions will enter inside the neuronal ends, or we can say axonal ends, in the cytoplasm of the axon. Why the calcium ions have entered? Because that nerve impulse have lead to opening of these ion channels. There is change in the voltage gated calcium ion channels, they open, right? What are these? These are voltage gated calcium ion channels. So voltage gated calcium ion channels have been opened. Calcium fuses with the synaptic vesicle. What is this? This is synaptic vesicle. And when the synaptic vesicle fuses with the membrane, it will release the chemical. You call it as neurotransmitter. Neurotransmitter. So the neurotransmitter have been released in this space, and the name of the space is synaptic cleft. What do you call it as? Synaptic cleft. And the entire junction is synapse. It's a chemical synapse. The entire structure is chemical synapse. The space between the two neurons is synaptic cleft. And the neurotransmitter is released in the synaptic cleft. Now, for example, we want to excite this neuron. So accordingly, a neurotransmitter will be released. For example, if I say the acetylcholine is released, and this acetylcholine, or we can say adrenaline, better say adrenaline. Adrenaline is a hormone and it is also a neurotransmitter. So here the adrenaline is released, it will bind to these channels, and these channels will lead to the entry of sodium ions. And whenever there is entry of sodium ion, that causes depolarization, and whenever there is depolarization, there is excitation. There is excitation, right? And now imagine a neurotransmitter is released, that is dopamine. What is it? Imagine that's dopamine. Dopamine is inhibitory neurotransmitter. Now if this one attaches, it will always attaches to potassium ion channel, and that will lead to the exit of potassium ions. And when potassium ions leave, that state is repolarization. And when there is repolarization, there is always inhibition. That is not excitation. During repolarization, it is going to be into rest state. That means it will not do anything, it will be inhibited. And in the depolarization, it will be in the excitation state. So here we learned that with the help of a chemical synapse, we can generate two types of responses. One is inhibition, one is excitation. For example, if I want to contract my muscle, I will be releasing an excitatory neurotransmitter. Imagine it is adrenaline, and that will lead to opening of sodium ion channel, and my muscle will get the nerve impulse and it will contract. Now, if I want to stop the muscle from contracting, my brain is sending dopamine or any inhibitory neurotransmitter, and my muscle relax, right? So this is how different types of neurotransmitter, they can lead to the different type of responses. That's a positive point of chemical synapse. What's about the electrical synapse? In the electrical synapse, there are two neurons and both are attached to each other, and in between them are present gap junctions. What are present? Gap junction. These gap junction can lead to bidirectional flow of ions, right? So from here, sodium ions or potassium ions, they can easily move in or outside. That can, imagine if these both are sodium ions, these both can be moved. And as a result, the wave of depolarization can move from one neuron to another, right? So what do you think? Will they be faster or this will be faster? If I say in the electrical synapse, there are just junctions present. What are these junctions? Gap junction. So if I say the stimulus have arrived on this neuron, if this one receives the stimulus, this can easily cause the depolarization of this one by sending the sodium ions. So simply, there is nothing to do. You don't have to fuse calcium ion with the synaptic vesicle, no release of neurotransmitter. They're just simple. And hence, they will be little faster. They will be little faster than chemical. But chemical one have a very important role, and that's a benefit. They can generate two type of responses according to the use of the neurotransmitter. So it's kind of a better one. Fine. But the faster one, they are the electrical synapses. So that's all about the synapses, guys. Let's move further. Let's move further and solve this question. The postsynaptic potential is always excitatory, always inhibitory, both excitatory and inhibitory, none of the above. So postsynaptic one can be both excitatory and inhibitory. We have done right now. So this potential that have arrived here in the postsynaptic neuron is also known as postsynaptic potential. Okay, so this postsynaptic potential, they can be excitatory as well as inhibitory. Fine. Next. Okay, let me write the answer. Answer is 3. Next, guys. When a neuron is not conducting any impulse, that is resting, the axonal membrane is comparatively more permeable too. For example, this is minus 70 millivolt. So this membrane is more permeable to more exit of potassium ions. So simple answer is 2. Next. Sodium potassium pump transport 3 sodium outwards and 2 potassium inward. 3 and 2, answer 2. Okay. So these type of questions are brainstorming question. That will make sure how much your concepts are, have been built nicely or not. The electrical potential difference across a resting plasma membrane is called the, very simple, what do you call it as? Electric potential difference across the resting plasma is resting membrane potential or resting potential. Next. Depolarization occurs due to influx of potassium ions, efflux of sodium ions, influx of sodium ion. Very simple. Whenever there is stimulus, it leads to opening of sodium ion channels. There is influx of sodium ions. Sodium ions enter inside. So influx of sodium ions causes depolarization, and efflux of potassium ions causes repolarization. Okay, all right.